research. At CBAF, scientists can advance our understanding of matter itself by probing the quarks and gluons in the atom's nucleus with beams of electrons from a superconducting accelerator. CBAF has an educational mission as well. The CBAF Science Series features scientists and engineers who are gifted at making their work not only informative, but fun. The Science Series is held once a month during the school year in the CBAF Center Auditorium. The presentation is live, spontaneous, and for the most part, unrehearsed.
It's my pleasure to introduce tonight's speaker, whom I've actually known for about 18 years. It's Dr. Saundra Oyewole. Dr. Oyewole grew up in Washington, D.C., where she attended public school and then went to Howard University and got a bachelor's degree in zoology. She then continued her education at Howard and at the University of Chicago, where she received a PhD in microbiology. Dr. Oyewole has been active in research and particularly active in teaching. She's been a professor for many years at Trinity College, which is a women's college in Washington. And she happens this year also to be working for the National Science Foundation, helping the government to decide how to award funds for people to do scientific research and work on science education programs. She picked for her topic today AIDS, the science, the impact, and is particularly hoping to answer questions you might have. So she may call for questions while she's speaking, and she will also want to engage in dialogue. She's planning to stop a little early so that you can ask her questions at the end of her talk. So please don't leave as soon as she stops talking, because I think that's when it will get particularly interesting. Anyway, please join me in welcoming tonight's speaker, Dr. Oyewole.
Thank you very much. It's really a pleasure to be here. And it is also a pleasure to be, I don't know, it's hard to call talking about AIDS a pleasure, but I do think it's something that's very important for all of us to be well informed about and to be able to share that information with others. As Bev said, I'm now working at the National Science Foundation, and in preparation for tonight, I had one of our computer specialists connect me with World Wide Web so that I could find out what the latest information available on the topic happens to be. And I told him that I wasn't sure about the audience, the age group, and exactly how I would pitch the presentation. And he thought about it and he said, but you know, you have the power to save a life by what you say tonight. I'm going to talk about the biology. I'll be talking about the virus, some of its features that have been discovered since in more recent years. I'll also be talking about the immune system, because the human immunodeficiency virus is a virus that attacks the immune system. Why is AIDS so deadly? It's so deadly because it affects the very system that usually protects us from infection. I've been interested in AIDS for a very long time. How long? 1981. Okay? Now, it looks to me like we have an older audience. I had watched a couple of videotapes of these presentations, and I had seen younger students in the audience. It looks like we have an older audience tonight. And I was thinking that many of those younger students would not have even been born at the time that the first hint of the HIV onslaught first came to our attention. And what I'd like to do is go back and look at the history, okay? And we don't have to go very far. And let me just run this down for you. On June 5th, 1981, the Centers for Disease Control, in its Morbidity and Mortality Weekly Report, described 5 cases. 5 young men treated at 3 different hospitals in Los Angeles, California, all homosexuals, all having an unusual form of pneumonia. And that pneumonia caused by a protozoan, Pneumocystis carinii. The reason that this was worthy of reporting in the CDC report was that it's very unusual to find cases of pneumonia caused by this particular organism. Most pneumonias are caused, there's bacterial pneumonia, and there are different varieties of viral pneumonia. But for a protozoan to be involved in the kind of damage to lung tissue that was being observed was remarkable. June 5th, 1981. What we find by July 31st, 1981, 26 cases of pneumocystis pneumonia. This time we're starting to see the appearance of a more invasive form of what had been thought to be a mild form of skin cancer, Kaposi's sarcoma. And so of those 26 patients, 10 had both pneumocystis pneumonia and Kaposi's sarcoma. So we're starting to see a linkage. No one has heard of acquired immune deficiency at this time. Acquired immune deficiency syndrome. The term had not been coined, the acronym had not been coined, AIDS. The fact that the earliest cases of this particular syndrome, why was it called a syndrome? Because at this particular point, they were seeing specific conditions, maladies associated with, in addition to the diseases that I've indicated, a depressed immune response. Okay? So we have an unusual form of pneumonia caused by a protozoan that usually is not implicated in cases of pneumonia. We've got a mild skin cancer becoming very aggressive. And by aggressive, I mean that it is a more metastatic, it is highly metastatic and causes, and is more likely to be fatal to the patient. Because of the association with the homosexual community, this syndrome was first called gay-related immune deficiency syndrome. Okay? One of the things that I want to say to you right now is that AIDS is not a gay disease. It's a disease that frightens me. How many of you are scared by AIDS? How many of you are scared of AIDS? Okay. How many of you think that your risk of contracting HIV is very small? Okay. Look at the person sitting next to you. Can you tell if that person is HIV positive? Absolutely not. The person sitting next to you looks like millions of, well, I shouldn't say millions. Well, if we do worldwide, and that's one of the things that I'd like to do now. I've been talking about AIDS for some time, and in fact, when I first started talking about it, the virus had not been named HIV, and we'll leave this on for just a short period of time. Acquired immune deficiency syndrome, the cause, human immunodeficiency virus. The virus was originally called The human T lymphotropic virus by Robert Gallo and his group, so many in the United States were using HTLV3 as the name of the virus associated with this syndrome. A scientist in France at the Pasteur Institute, Luc Montagnier, isolated a virus that was associated with this syndrome, and he called the virus lymphadenopathy-associated virus. There was another group, another research group in this country that isolated a virus associated with the syndrome, and they called their virus AIDS-related virus. There was a lot of confusion within the scientific community for a number of years, and some of you may be aware of the politics. I mean, there in fact is politics within science. There was some tension between the American group and the French group as to who in fact discovered the virus first. More recently, Gallo has confirmed that the virus that he isolated was actually a virus that was isolated from tissue that had come from the laboratory in France. We're going to look at the target cell. The target cell of HIV is the T4 lymphocyte. We'll talk about that lymphocyte. How is the virus transmitted? Now, this is an interesting part of the history. This is a medical mystery. You had individuals with an unusual set of symptoms. What's going on? I'm a microbiologist, and I attended the first meeting on AIDS at the general meeting of the American Society for Microbiology. There was standing room only. We were all curious about this new disease. And at that particular time, there had not been a direct link made between the virus, HIV, and the set of symptoms, the syndrome. And so many thought perhaps it was a virus. Others thought perhaps it was a bacterium. The question, why gay men? What is it about their lifestyle, their exposure, that would lead them to be more susceptible? And I think we were all, I shouldn't say all, but some of us were less informed about sexual practices within the gay community than others. But once it became revealed that this virus is transmitted by certain body fluids, specifically blood. Another piece of the puzzle, gay males, pneumocystis carinii, Kaposi sarcoma, and then we found cases of the syndrome among hemophiliacs. What do you know about hemophilia? Their blood will not clot, okay, because they are missing a clotting factor. The therapy for hemophilia is to introduce exogenous clotting factors. Those clotting factors were extracted from blood, donated blood, okay? And because no measures were taken that would have eliminated or destroyed a virus, any virus, when these individuals were given the clotting factors, they were also being given the HIV virus. That was an important clue. Again, a medical mystery, a very important clue that perhaps whatever it is that causes AIDS is found in blood. But still, why gay men? What we now know is that during anal intercourse, there are tears in the anus, usually, and that makes it another fluid in which the HIV virus has been isolated. The HIV, I shouldn't say HIV virus because the V, why? The V stands for virus, okay? The HIV is also found in semen, okay? So we have HIV in that particular body fluid entering the blood through lesions in the anus. Okay? Now, we also found very early on in the history of AIDS in this country that people were contracting the virus by blood transfusions. It was a major discovery that Luc Montagnier and Robert Gallo made when they were able to demonstrate, and this could have been coincidental, that in a high percentage of the cases of AIDS that they studied, they also found what Gallo called at that time HTLV3 and what Montagnier was calling LAV. So there's a connection there. But is it the right cause and effect relationship? Okay? So the mystery continued, but do we have to wait until we have all of the scientific information before we act? You need to understand that the concept of contagion was understood before the age of microbiology. People understood that if someone had a certain disease, and there were a set of contagious diseases, that it was a good idea to stay away from that person until they appeared to be well. Okay? So the concept of contagion and quarantine preceded our understanding of the germ theory of disease. Okay, we've got some indication that there's an association between HIV and blood. Blood transfusions are life-saving. It is important for you to understand, I feel a need to say this at this point, you cannot contract the virus by donating blood, okay? But if in some way blood carrying HIV, you can understand that I've used HTLV3 and had to add virus to it for many years, and so I'm making the transition. Actually, a committee, an international committee, decided that it's time to have one name for this virus after it became very clear that we were really looking at the same virus. And then, of course, human immunodeficiency virus was the name selected. So, sexual contact, body fluids, contaminated blood or blood products, so blood transfusion, blood product, that information came from the observations of the occurrence of AIDS in hemophiliacs. And it is also possible for the virus to be transmitted vertically. What does that mean? From mother to neonate. And there is a very tragic story. In 1982, again, going back to the history, there were reported cases of heterosexual couples transmitting the virus. And so we're now moving away from thinking of AIDS as a gay disease. There was a family the Burkes, the woman, the wife, contracted AIDS from her husband who had hemophilia. Okay? How did he get the virus? From a contaminated preparation of the clotting factor. So he had AIDS. Well, he was infected with HIV, transmitted that to his wife through heterosexual intercourse. She became pregnant and transmitted the virus to their son. Okay? What I have said to people I care about is that AIDS is not about trust. It's not about how well you dress. It is about a virus, and that virus can take you out. And you need to respect it, and you need to think very carefully about your behaviors. There is no cure for AIDS. There is no vaccine. The only weapon, abstinence is, in terms of sexual transmission, abstinence is, in fact, the safest procedure. Okay? Obviously, abstinence is not going to necessarily protect you from contaminated blood. We are very fortunate that as of 1985, our blood supply has been well controlled and tested for HIV. And although it is always possible for a mistake to be made, we can feel very comfortable that it is very unlikely that any of you will contract HIV. Now we're talking about the virus through contaminated blood. We can't say the same if you're not in this country. Okay? So the blood supply is protected. But the other methods by which one can, tell me of a method of contracting HIV that I haven't mentioned is up here. IV drug users. What if you're using metabolic steroids? Might you still transfer HIV from one person to another? Does it have to be heroin? No. A blood contaminated needle. So it's very important, especially for young people, to understand that it is the needle contaminated by blood of a person carrying the HIV virus, or HIV, it's hard, that is actually the agent of exchange. It is important for you to understand that this is a virus that is transmitted by direct contact, body fluid to body fluid. Let me hear from you. Can you get AIDS by sharing a room with someone who has? No, good. Very good. What about using the telephone with someone who is HIV positive? No. Okay. So it's important that we understand that there are specific ways by which the HIV virus is transmitted from one person to another. I want to show you a modified transparency. As I said, I've been talking about AIDS for a number of years now. In black, you see the data that I used to use. In the early 80s, actually mid-80s, 100,000 cases worldwide. Now you need to understand what this is. These are reported cases. In this country, we were paying careful attention. We had a system whereby any cases of pneumocystis carinii, Kaposi's sarcoma, had to be reported to the Public Health Department. And the Centers for Disease Control in Atlanta was keeping a record. We also had an assay, a serological assay for HIV, which allowed us to determine, or at least in some cases, I have to talk about the limitations. If there are antibodies present in the blood to HIV, there are techniques for detecting them. Can a person be HIV positive? Can a person be infected with HIV and not be HIV positive? Can you have the virus and when tested by the available tests that we have? Yes. Yes. What does that mean? It means that you don't always know. Okay, let me put it this way. Just because, if you've engaged in a risky behavior, or risky behaviors, and you've decided, oh shucks, maybe, let me go get tested. And you get tested within, say, a 2 or 3 week, maybe even 4-month period of engaging in that risky behavior, and your test comes back HIV negative, and you go, phew, okay, maybe I'll change my behavior, maybe I won't, but I'm safe. You may not be safe, because it does, in fact, take some time for the antibodies, which are the basis of the HIV assay, to develop within the body. Okay, so very quickly, 100 cases reported worldwide in the mid-80s, actually. In mid-94, actually, 985,000 cases. If we just look at the US, 33,000, 200,000 in 91. And if we drop down and look at the percentage of homosexual males among those in the US with AIDS in the 80s, 70 to 75% homosexual, the 90s, 48%. What does that mean? It means that the incidence of AIDS in the heterosexual community is increasing. Okay? And you need to know that the rate of increase is greatest among women, particularly Black and Hispanic women. 1992 estimate, more than 1 million Americans infected with HIV. And we have to make the distinction between HIV infection and AIDS. HIV infection indicates, simply means that the virus has entered the body. AIDS is actually a set of symptoms that will occur in advanced stages of the viral infection. And I've got to quickly move on. I think I've hit on some important things, but now it's time for us to talk a little bit about the virus, okay? Here we have an artist's representation of the HIV virus, okay? A virus is not a living organism. It is a microorganism. It sounds like a little bit of a conflict there, but it's not cellular. It's acellular. It's an obligate intracellular parasite. It cannot, what that means is it cannot live outside of cells, okay? This is a representation of the AIDS virus based on scientific evidence. This virus is an RNA virus, and so here we have represented the genome of the virus. The genes of the virus are in the form of RNA, and in fact, we have 2 copies of that RNA. The other thing that we have that is unique, have represented here, is the enzyme reverse transcriptase. I have to keep my hand off the microphone. Reverse transcriptase is an unusual enzyme found in a subset of viruses called retroviruses. They are identified by the presence of this particular enzyme. We also see represented here that this virus consists of a nucleic acid core, which happens to be RNA, ribonucleic acid, with a protein coat. And in addition to the protein coat, we see that there are spikes on the virus. And we also see represented what is called the envelope. The envelope is actually derived from the infected cell. Okay? And let me show you how that happens. Okay? The virus enters the cell. Okay? First, there has to be attachment of the virus to receptor sites. The target cell for HIV is the T4 lymphocyte. The T4 lymphocyte has a receptor on it called CD4. Okay? CD4 and one of the glycoproteins found on the surface of the virus will combine. CD4 is actually the receptor for the virus. In order for a virus to enter a specific cell, the virus, some part of the virus, has to interact with the surface of the cell, and subsequently the virus will enter. And so what you see are a sequence of events associated with a viral infection. In this particular case, we're looking at HIV. The virus attaches to a CD4 receptor, so this represents a lymphocyte, a T4 lymphocyte, which you know is round, but this is the artist taking liberties. So we have the viral RNA. What is unusual about this particular class of viruses is that because of the presence of the reverse transcriptase, this virus is able to direct the synthesis of a piece of deoxyribonucleic acid, DNA, that is complementary to the RNA strand. Now, in humans, DNA is the genetic material. In this particular virus, however, the genetic material is in the form of RNA. What you see is that after the strand of DNA that is complementary to the strand of RNA is synthesized, the RNA is destroyed, and we now have a double-stranded DNA molecule that really represents the genes of the virus. Why does it sometimes take 10 years for full-blown AIDS to develop? This virus, this DNA, is able to integrate into the host chromosome and form a provirus. These are viral genes in the form of DNA integrated. And what I mean is they've been joined covalently. The viral genes have been joined covalently to the host chromosome. And in that form, the virus can remain dormant for a number of years. We don't know what activates the provirus and ultimately what then leads to the production of virus particles. So here we have represented a reflection of what happens in viral replication. Viruses are acellular. They don't grow. What happens with viruses in viral replication is that the parts are made and then they assemble. Okay? So here we see the fact that we see represented proteins of the viral coat. We also see the glycoproteins having been synthesized, coming together within the cytoplasm of the cell. We see here the assembly, the partial assembly of the virus. And we see also represented the fact that this virus leaves the cell by a process which we call budding. And as the virus leaves, it picks up part of the cell membrane. So the retrovirus has an envelope. And that envelope is derived from the infected cell. I have a transmission electron micrograph of a normal human lymphocyte. Do you know what a lymphocyte is? A white blood cell found circulating in the blood. This is a normal cell. And now I want to show you what happens to that cell once. Can this cell do anything for you? No. Okay? Now, the nucleus is totally destroyed. These are virus particles. These are the virus particles, and this is what is left of the cell. This is no longer a functional cell. This is one of the cells that is supposed to protect you. And if we move now to a look at the immune system, there are two basic types of lymphocytes. There are B cells and T cells. The B cells are the cells that are responsible for making antibody. Antibody molecules are proteins. They bind to antigens. What are antigens? They are foreign substances. We're full of them, okay? The best antigens happen to be proteins of a molecular weight greater than 10,000 Daltons. Without going through, what this shows you is that lymphocytes are really derived from cells found in the bone marrow or the fetal liver. This is during embryonic development. We then find a lymphocyte stem cell. Some of the cells that are derived from this stem cell become B cells. Others become T cells. T stands for thymus. And the reason they're called T cells is that it's very clear that these cells that are called T cells are processed by the thymus gland. The B cells are called B cells because of their similarity to cells found in chickens that are modified in a structure called the bursa of Fabricius. We don't have one of those, but because of the similarity of these cell types, these are called B cells. B cells produce antibodies. T cells are the primary target of HIV. All right. So why is it so devastating when T4 cells are destroyed? A term that I haven't used yet is T4 helper cell. I don't expect you to follow everything. I'm going to try to lead you through this. Here we have the helper T cell. This T cell, there are different antigenic types. The specific T cell that is infected by HIV is the one that has the CD4 receptor. That cell stimulates B cells, and it also stimulates cytotoxic T cells. B cells give rise to plasma cells, which secrete antibodies. B cells can also give rise to memory B cells, Memory B cells are responsible for protecting you in case you are exposed to the same harmful antigen at some later point. They are the basis of immunization in that an immunogen is injected. You've all been immunized, well, maybe not. I've been immunized against smallpox. How many of you have been immunized against smallpox? Okay. You were given an immunogen which allowed you to develop antibodies, protective antibodies against the smallpox virus. So if you at some later point in time were to be exposed to the smallpox virus, you would already have B lymphocytes, memory B cells, that would start to attack that invading organism more quickly, and so you would not develop smallpox. So that gives you some idea of the role, without going into a great deal of detail, the connection between helper T cells, B cells, and antibody production. What you need to understand is that not all antibodies are protective, and one of the difficulties in AIDS research has been the development of a vaccine that would stimulate the production of protective antibodies. Just any old antibody is not necessarily going to prevent the disease. Okay. Let me show you some diagrams that indicate how antibodies function. Antibodies can bind to a virus. Because antibody molecules are multivalent, they can bind to more than one virus particle and form aggregates of those particles. And once those aggregates are formed, then phagocytic cells are able to engulf those aggregates and destroy them. Why does the immune system not destroy HIV? Why? It's now time for questions. We've got 10 minutes. I know that a number of you will have to leave at 8, and I do want to have some chance to answer questions and to share information. Why is it that, this is my question to you, why is it that HIV is not eliminated by antibodies the way other infectious agents are?
Maybe the antigens aren't strong enough.
Maybe that's actually, okay, maybe the antigens aren't strong enough. Remember, I made the point that not all molecules are antigenic, are good antigens, in other words, will stimulate antibodies. Let me define. Antigens are foreign molecules. Antibodies are molecules, proteins, that are synthesized in response to an antigen that then combine specifically with that antigen. Some proteins are more effective than others. There is another piece to it, though.
Because the AIDS cell uses the T4 wall for its own wall, so
What I'd like to remind you of, okay, what's on that diagram? The key, the provirus. When the AIDS virus is in the provirus state, it's invulnerable to antibody. Can't reach it. There's another piece. You mentioned the cell, okay? And let me develop your answer a little bit more.
Because when it procreates inside the T4 cell, it uses its wall for its own, like you said.
Okay, what he's suggesting is that because of the envelope, the fact that much of what is exposed on the exterior of the virus is host material. In fact, though, there are obviously the spikes do extend beyond that envelope. But that's an interesting point. Any other suggestions? Yes. I'm ready. I'm open to questions, so let's hear them, okay? I don't want to ask all of them. Yes, here?
This isn't a suggestion, but recently on CNN, I heard that there was a baby born about 5 years ago who tested HIV positive. And now that 5-year-old child no longer has
That's right.
HIV. So what does that
You can believe that that kid is going to be studied. There's no I mean, but of course there are ethical considerations. But this is an amazing observation. We have no answer. We don't know why. So it'll be interesting to follow this child without intruding too much into the child's life, to see, in fact, whether there is anything unusual about the immune system of that particular child that distinguishes it from others who have been diagnosed as HIV positive. Yeah, that's good. Yeah. Well, there was a hand over here.
What is AZT and what effect does it have on AIDS or HIV?
AZT is azithromycin. It used to be and probably still is, the drug of choice for treatment of individuals who are HIV positive. AZT attacks the provirus state of the virus in that it interferes with DNA synthesis. But the downside is that there are a lot of side effects to AZT treatment. And so many who are HIV positive have decided not to use it. But others have found positive results. So we're still looking for better drugs, but many are being treated with azithromycin. I have to repeat. The question was, what is AZT?
What about making a virus that does the exact opposite of this? That goes in and breaks down this, like with the same attraction, goes in and breaks down the provirus and does the reverse. Can you create viruses like that?
The question was, would it be possible to create a virus that would reverse the effects of HIV? There would be a Nobel Prize in that. You are aware of the techniques of DNA recombination, the fact that it is possible to manipulate genetic information. It is possible because of the way that viruses assemble. I don't think that anyone is necessarily devoting a lot of time to that strategy, but sort of related, there are strategies that involve interfering with the binding of the virus, the GP120 of the virus and the CD4 of the cell. Okay, you have a question? Yes.
Yeah, if they were to cure the cure for AIDS,
yes,
wouldn't they use like a cell that was sort of steroid type, but not in the sense that it would give you side effects, but it would be strong enough to destroy the T cell without breaking any of your white blood cells?
Okay, that was going to be hard to repeat, but I think the idea is that if you're going to try to develop something that protects against AIDS, wouldn't that entity have to be less harmful than HIV and not destroy the cell? Well, certainly. Certainly. Yes, in the back.
My question kind of relates to what was said earlier. Would it be possible to have something that attaches to the CD4 receptor so that HIV cannot attach itself?
There are labs. The question was, would it be possible to have something that attaches to the CD4 receptor and blocks the binding of HIV? That is a strategy that is being studied in the laboratory. Yeah. Because if you can prevent the virus from getting into the cells, then you can prevent replication of the virus. Other questions? Yes.
How long from initial contact till people start showing symptoms of AIDS? Like, what is the longest known case?
How long between initial contact and the appearance of acquired immune deficiency syndrome? It varies. And in some cases, you can't really determine when, in fact, the person was exposed. Right now, we're saying 10 years, but I do believe there's someone who's gone longer than that. Yeah, but the average is about 10 years. Yeah.
I heard somewhere as one of the possible cures, they actually boiled the blood of the people and something like that. And they did something to destroy them.
The question, this I've not heard of, that in order to...
I guess it was to destroy the virus.
Oh, to destroy the virus, blood was boiled. Well, depends on if you are using that blood as a source of clotting factor. You have to be it depends on, I'm not familiar with that particular procedure. It very much depends on how that blood is going to be used. If you want to use that blood in transfusion and you've destroyed the proteins that are necessary for that particular blood to help the person receiving the transfusion, then that wouldn't work. But if you were trying to isolate some blood product, heat might be a way of, but that would be controlled heating that might destroy the virus, but yet not destroy the products that you were trying to purify, isolate.
I don't know if you already answered this, but can it be transmitted through oral sex?
Listen, don't move. Can HIV be transmitted through oral sex? Absolutely. Absolutely. Yes. Oral sex, anal sex. What are the other kinds? Yes. So it's very important. I would recommend that you abstain from sex. But if you are sexually active, you need to be monogamous. You need to be involved with a single individual who's only involved with you, who's never been involved with anyone who has contracted, who has been exposed to HIV. How are you going to do that? Okay? So if you are having sex, you need to use a latex condom. Okay? The safest way is not to have sex at all. Cleaning up people who are bloody, you use gloves.
Leave them.
No, you don't leave them. The other thing, is that actually it was my nephew who suggested that maybe the rap singer who recently died of AIDS, Eazy-E, yeah, I knew I forget. Eazy-E deserved to die because he was a gangster rapper and he talked about really awful things. No one deserves to die of AIDS. And one of the bumper stickers that you'll see around, fight AIDS, not people with AIDS. Okay?
I think that may be a good note to end on.
Yeah.
Dr. Oyewole is delighted to take questions sort of privately, and a lot of people have to leave.
Yeah.
And please give to some CBAF person standing around your evaluation forms, especially with ideas for speakers for next year, and thanks for coming.
It's just as well that I didn't have my slide.